Today we will discuss about this adaptive cell transfer therapy for this tumor infiltrating lymphocyte. So in this case, the idea is to use autologous TIL or the tumor infiltrating lymphocyte. This means that this lymphocytes will be used from the same patient. So idea is shown in this figure on the left hand side, where what they will do is they will take out the tumors from the patient and culture them into different wells. First they do the fragment of these tumors and culture them into different wells. So this tumor infiltrating lymphocyte, the name itself suggests that tumors is infiltrated with lymphocyte, and lymphocyte are the kind of immune cells that can kill the tumors. So the idea is that when they fragment the tumors and culture them in the individual culture them in the different wells in the presence of IL-2, some of the wells in which the lymphocytes are able to target the tumors will outgrow the tumors and ultimately kill the tumors. So after 2 to 3 weeks of culturing them in the presence of IL-2, here the concentration is mentioned here the concentration mentioned as 600 IU per ml IL-2, and so after 2 to 3 weeks they will check each well for this tumor recognition. And the idea is that once the lymphocytes scaled up, they will kill the tumors completely, so they will not see any tumor signs in some of the wells, and those wells will be used to scale up to this 10 raised to power 10 cells, as those wells will be considered as the lymphocytes that can target the tumors, and those tumors can thereafter those lymphocytes or the expanded tumor infiltrating lymphocytes can then be reinfused post lymphodepletion. And why this lymphodepletion required, that we have already discussed in our previous videos, in our recently published previous videos, where we have understand that the lymphodepletion or the immune suppressions is necessary to avoid the excessive immune reactions that might cause the cytokine release syndrome, and those cytokine release might cross the threshold value of the patient depending on the various factors such as the tumors size, tumors quantity. And in order to suppress those CRS levels, what the idea the people used is to suppress the immune system, is to suppress the immune system of the host, and because immune system or exogenous or this the natural immune system of the host, so that the cascade effect will be suppressed and the CRS levels will remain within the limit. And there is if we see the figure on the right hand side, we can see the lymphodepletions prior to T cell transfers, and thereafter followed by the immune reconstitutions. So here what it is, they first do the lymphodepletions using these two reagent, that is cyclophosphamide at around -7 to -5 days, or for the 2 days before the 2 days, and thereafter they use this fludarabine for 5 days, and after that they are able to see a low amount of white blood cells or neutrophil counts and the lymphocyte counts. Then they did the T cell transfers along with IL-2, and after around 5 days they reconstitute the immune system within the host, or reconstitute the immune cells within the host, and we can see the further increase in the immune cells, that is white blood cells, absolute neutrophil count, absolute lymphocyte remains at a lower level, because in that patient lymphocytes amounts will be seems to be at this level only. So, and here we can see that the reconstitutions done just after 5 days, whereas in the previous videos where we checked this adaptive cell transfer therapy for CAR T cells, we have seen that the tumor, we have seen that this tumor immune reconstitution is done after 28 days. So it varies based on what kind of adaptive cell transfer therapies patient is undergoing. So that's all for this video. If you need to understand more about this approach, this ACT approach in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.